Hello students, myself Bhanu Solanki, Senior Faculty Sri Chaitanya. Today we are going to discuss one more chapter, Principle of Inheritance and Variations, means genetics. What are the areas in this chapter that in these last moment of your preparation we should pay attention? Today we are going to explore. So come on. In the first section there the two things that you should keep in your memory. One, what is the inheritance and what is the variation? So transmission of the characters from the parent to progeny, that is called heredity, and inheritance is the process by which the characters are passed on from parent to progeny. It means you can say that the inheritance is the basis of heredity. Passing on of the characters from parent to progeny called heredity, and the principle, the basic mechanism behind it is the inheritance. Second part is the variations. Variations means differences among the individuals of the same species, that is called variations. So variations was hidden into the sexual reproduction, it we know since the beginning. The exploitation of the variations that were naturally present in the wild population of the plant and animals to selectively breed and select the organism that possess the desirable character. Here there, this example of the Sahiwal cows developed in Punjab should keep in your memory, and how it was developed you must remember by the selective breeding and domestication. Artificial selection and domestication were the two methods that were applied and used during the preparation of Sahiwal. Then in the classical part of the genetics, their Mendel related some informations are very crucial. First of all, Mendel did experiment on garden pea, means Pisum sativum, for how many years? For 7 years. In during which years? Then you should keep it memory in from 1856 to 1863. These 7 consecutive years he did work and proposed the laws of inheritance of the living organisms. During Mendel's investigation into the inheritance pattern, it was first time that the statistical analysis and mathematical logics were applied to solve the problems in biology. How, or you can say why Mendel was successful? His experiment had a large sampling size. Very important point. One of the reason behind the success of Mendel, large sampling size, which give them greater credibility to the data that he collected. Also, confirmation of his inferences from the experiment on successive generations of the test plant, means two things are here there highlighted. One, large sample size. Second, confirmation of the results for the up to F2 and F3 generation was very crucial, and large sampling size was giving greater credibility to the data, and when the crosses were going on till the F2 and F3 generation, then the consistency of the pattern of inheritance get reflected properly. So it is also, these were the two reasons that why the results of Mendel's rules of, were pointed to be the general rules. It proves that his results proved to be the general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated character into the garden pea, and for the study of the characters in garden pea, he selected 14 pure lines or the true breeds. Group of homozygous organisms is created by the repeated selfing is known as pure line. So Mendel selected 7 characters, it means Mendel created 14 pure lines. Tall plant and tall plant and dwarf plant, 2 pure lines. Round seed, wrinkled seed, another 2 pure. Seed color, yellow color dominant, green color recessive. Be careful, seed color means, for Mendel, seed color means cotyledon color. When he used seed color, so he assumed to the cotyledon's color as the seed color. Then flower color, violet was dominant, white was recessive. Pod shape, full means Inflated was the dominant character, constricted was the recessive. Pod color green was dominant, yellow was recessive. Flower position axial yani axillary position was dominant and terminal position was recessive. And tall plant, dwarf plant we have already discussed. So these were the 7 contrasting characters by using 14 pure line breeding true varieties of the plant he selected for the experimental work. This table you have to keep in your memory by heart because it may be the basis of various questions. Suppose they can ask that which of the following character will express in both homozygous and heterozygous condition. We know that dominant character express in both homozygous and heterozygous condition, or vice versa. Which of the following character will express only in homozygous condition? It means you have to select the recessive character. So this table please keep memorize. Then inheritance of the one gene means monohybrid cross. In the monohybrid cross, he take one character under consideration: height, shape of the seed, pod color, pod position, flower color, seed color, many. Every time one character he consider and he did his experiment. When he did a cross between a pure line tall plant with a pure line dwarf plant in F1 generation, Mendel observed that all plants of the F1 generation were tall plants. Like one of his parent, none were of the dwarf. He made a similar observation for the other pairs of characters also. I said already, for all 7 characters he did this experiment. F1 always resemble either one parent and that the trait of the other parent was not seen in them. Mendel then self-pollinate to the F1 of F1 tall plants or F1 dominant character showing plants, and his surprise found that in F2 generation both of the parental trait means tall and dwarf express. The proportion of the plant that were dwarf were 1/4 and in the F2 generation, while tall were the 3/4. It means tall is to dwarf were arising in the 3:1 ratio. When he applied the same thing on other character, the same pattern comes, means F1 generation only one trait express. In F2 generation both dominant and recessive trait express in 3:1. For all 7 character it was consistently happened. It happens likewise, so on the basis of which he said that based on the observation, Mendel proposed that the something was being stably passed down unchanged from the parent to offspring through the gametes over successive generations. He called these things as the factor. At the time of Mendel, there was no idea of the gene, so he was using the word factor. Now we can call them as a gene also. Therefore, the unit of inheritance is the gene. Very important point. The unit of inheritance is gene. They contain the information that is required to express any particular character. Genes which code for a pair of contrasting traits are known as alleles. It means you can say that the alleles are slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letters are used to denote the dominant gene and the recessive small case or the small letters are used to denote the recessive trait. It means capital T would be the pure line tall, capital T small t would be the heterozygous tall, and small t would be the recessive tall. As Mendel found the phenotype of the F1 heterozygote was the capital T small t to be exactly like that of the capital T parent into the appearance, means heterozygous tall were as tall as the homozygous pure line tall. He said that the tallness is the dominant factor while the other factor small t was the recessive factor. In this case, capital T denote to the dominant over the small t. So it is the Punnett square where there it is showing that homozygous tall crossed with the homozygous dwarf. In F1 generation all the plants were tall plant, capital T small t, capital T small t, capital T small t, capital T small t. When they allow for selfing, so you must remember that the tall is to dwarf obtained in 3:1 ratio phenotypically, and if genotype is concerned, homozygous tall 1, heterozygous tall 2 and homozygous dwarf 1, means 1:2:1 is the recessive, I mean to say genotype of this cross that they obtain. The Punnett square shows that the parental tall, capital T and parental dwarf, the gametes produced by them are only tall and dwarf, while the capital T small t progeny, when become parent and they produce the gamete, 50% gametes will having the capital T and 50% have the small t. Capital T have 50% chances to be pollinate X with the genotype capital T. Means it is showing the probability. In this manner, we can say it is a kind of a binomial, means capital T crossed with small t, we will obtain capital T and small t. When capital T small t self-pollinate, it means capital T small t crossed with the capital T small t. It means A square plus 2AB plus B square is the binomial. It means capital T equivalent to the A square, capital T small t is equivalent to the 2AB, and small t means B square, like that. The binomial, it is following 1 by 4, 1 by 2 and 1 by 4 is the ratio of capital T small t and small t. So in this manner, Mendel self-pollinate to the F2 plant and found that the dwarf F2 plant continued to generate the dwarf plant when into the F3 generation as well as in F4 generation. He concluded that the genotype of the dwarf was homozygous. What do you think that he would have got the half and he self-pollinate to the tall plant of the F2 generation? Yes, it is interesting. Pure line tall will give rise to the tall, but the heterozygous tall will give rise to the tall as well as dwarf both. Mendel crossed tall plant of the F2 generation with a dwarf parental plant that he called test cross. Means test cross is a cross in which we can find out genotype of the dominant phenotype showing organism. As I have already said that recessive phenotype always expressed only in homozygous condition. It means for the recessive phenotype showing organism, we need not to do any kind of a test cross because it is clear. But dominant phenotype showing organism may be homozygous dominant or may be heterozygous dominant. To find out it that whether they are homozygous dominant or heterozygous dominant, there is a provision of a cross, and that cross is known as test cross. Yes, here that it is showing that the dominant phenotype showing plant, we don't know genotypically, it is a capital T small t or capital T genotype is not known. Now to know it, we will cross it once with the recessive parent, and if along with the cross with the recessive parent, if all flowers are violet, it means that the dominant phenotype showing plant were the homozygous dominant. In contrast, if after crossing with the homozygous recessive, we obtain tall plant as well as dwarf plant, means violet flower as well as white flower in 1:1 ratio, so it means the dominant phenotype showing plant was the heterozygous plant. This is called test cross. Based on the observations on monohybrid crosses, as I said already that he did experiment on all 7 characters, Mendel proposed two general rules to consolidate his understanding of the inheritance in monohybrid crosses. Today these rules are called principles of inheritance. First law of inheritance, that is popularly known as law of inheritance, and the second law of inheritance, that is popularly known as law of segregation. What law of dominance is saying? Law of dominance is saying that the characters are controlled by discrete unit, and these discrete units are known as factors, yani ki genes. Factors occur into the pair because only capital T or only small t, we cannot say that what kind of a phenotype will arise. So characters occur in pairs. In a dissimilar pair of a character, in a dissimilar pair of a factor, one member of the factor dominates while the another remain hidden. Means one of the factor will express and another one will remain hidden. The factor which get expressed phenotypically is known as a dominant factor, and the factor which not expressed in F1 generation, that is known as a recessive factor. So there are 3 points that indicate to the law of dominance, that the characters are controlled by the discrete units called factor. Factors occur in pairs, and in a dissimilar pair of factor, one factor dominate over the another factor. This is collectively called law of dominance. The law of dominance, very important point, the law of dominance used to explain the expression of only one of the parental character, only one of the parental character into the monohybrid cross in F1 generation, while expression of both of the parental trait into the F2 generation can be explained by the law of dominance. Expression of only one of the parental trait in F1 and expression of both of the parental trait in F2 generation can be explained by law of dominance. Second point also, phenotypic ratio 3:1 can also be explained by the law of dominance, because if law of dominance is not there, then the 3 plant cannot to be a tall. If 3 plants are tall, out of them 2 are heterozygous tall, and 2 heterozygous can be tall only in one condition, when capital T dominate over the small t. So 3:1 is possible because of the law of dominance. Hence, law of dominance is explaining to the 2 things. What? Expression of one of the phenotype in F1 and expression of both of the phenotype in F2 generation can be explained by law of dominance. Similarly, 3:1 phenotypic ratio into the F2 generation can also be explained by the law of dominance. Then law of segregation. This law is based on the fact that the allele do not show any kind of blending. It means both capital T and small t remain together, but they do not mix together, they do not blend together. That both of the characters are recovered as such into the F2 generation, it means they are not blending. Though one of these is not seen into the F1 generation, even after that they do not blend, and in F2 generation both recover. Though the parent contained 2 alleles during the gamete formation, the factor or the allele of a pair segregate from each other such that a gamete receive only one of the factor. Means in the F1 generation, where both of the factors are present together, they do not blend or mix with each other, and at the time of gamete formation they become separate. So one gamete receive only one allele of the each gene pair. Because one gamete having only one allele of the each gene pair, that is why this law of segregation also known as law of purity of gamete. Because in the F1 generation, when it take part in gamete formation, both of the allele become separate from each other, it is called law of segregation. Because of this separation, each gamete will receive either of the allele, so it is also called law of purity of gamete. This is one of the most crucial law of inheritance, and it is also said to be the universal truth of the biology or the generalization of biology. But there I must say that 3:1 phenotypic ratio in F2 generation is also because of segregation. Means 3:1 ratio in F2 generation can be explained by law of dominance as well as by law of segregation. Without law of segregation and without law of dominance, F2 phenotypic ratio 3:1 is not possible. Law of dominance have some exceptions, but law of segregation do not have any kind of a exception. One more fact I would like to discuss with you. In the anaphase 1 of the meiosis, when homologous chromosome becomes separate from each other, at that time the alleles become separate from each other. So you can say anaphase 1 of the meiosis is the cytological basis of law of segregation. In law of segregation, there is no exception, but in law of dominance, there are 2 important exceptions. One is known as incomplete dominance and second is known as codominance. Look at the incomplete dominance. When he did experiment on flower color in dog flower, means snapdragon or also called antirrhinum, is one of the best example of incomplete dominance. In a cross between a true breeding red flower plant and a true breeding white flower plant, the F1 capital R small r, red flowered plant it denote with the capital R, and white flowered plant it denote with the small r. When F1 generation we will obtain, all the plant would be capital R, small r. Phenotypically, all these plants were of a pink color. While what Mendelism said, what law dominance said, that F1 exhibit only one of the parental trait. While here, F1 is not indicating to any of the parental trait. Instead, it is showing incomplete expression of the red color due to the incomplete presence of the white color. Means both of the phenotype blend together. So F1 is an intermediate of both of the parental trait. When this F1 was self-pollinated, the F2 resulted into the red flower as well as pink flower as well as white flower. Means 1:2:1 is the phenotypic ratio that we obtain in F2 generation. It is unlike Mendelism where their F2 generation ratio was 3:1, phenotypic ratio I am talking. Genotype is same in both dominance as well as in incomplete dominance, 1:2:1. But phenotypic ratio in Mendelism was the 3:1 and here it is a 1:2:1. In case of incomplete dominance, F2 phenotype and genotype are same. So this is the case of incomplete dominance, means F1 exhibit intermediate phenotype of both of the parental traits. Why it happened likewise? Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some are recessive? To tackle these questions, we must understand that the gene does, every gene, as you know by now, contain the information to express a particular trait. In a diploid organism, there are two copies of the each gene, means a pair of alleles. Now, these two alleles need not always be identical, means they may be homozygous or they may be heterozygous. Let us assume a case that the normal allele capital T is producing a normal enzyme that is needed for the transformation of a substrate into the product, and when product will form, then the phenotype will appear. But theoretically, suppose this gene get modified, means get mutated. If the capital T get mutated, so it theoretically there are three possibilities, that it may produce a normal or less efficient enzyme, it may produce a non-functional enzyme, or it may produce no enzyme at all. If a modified gene is producing normal enzyme or less efficient enzyme, so it means it is capable to transform the substrate into the product. It means in this case also dominant phenotype will express. But if a non-functional enzyme is there or no enzyme is there, so it means the substrate will not transform into the product. Consequently, the phenotype will not produce. The phenotype is not produced, that means altered phenotype arise, and to whom we are calling recessive. So in the first case, means when normal and less efficient enzyme is forming, in the first case, the modified allele is equivalent to the unmodified allele. Why? Because it will produce the dominant phenotype. The result in the transformation of the substrate into the product. Such equivalent pairs are very common, but if the allele produces non-functional enzyme or no enzyme, the phenotype may be affected. Phenotype will only be dependent on the functioning of unmodified allele. Unmodified means functional allele. It means talls are tall because of the presence of capital T, and dwarf are dwarf due to lack of capital T. What author is writing? That the phenotype will only be dependent on the unmodified allele. Once due to presence of unmodified allele, and second time due to the lack of unmodified allele. Another exception of the law of dominance is given here under the title codominance. It is also exception of law of dominance. Till now we were discussing crosses where the F1 resemble either of the two parent, means dominance, or it was intermediate of both of the parent, yani ki incomplete dominance. But in the case of codominance, F1 generation resemble to both of the parental traits. Means now there are three conditions. F1 exhibiting one of the parental trait, means dominance. F1 showing intermediate phenotype of both of the parental trait, means incomplete dominance. And if F1 exhibiting both of the parental traits simultaneously, so we will call it codominance. So F1 resembles both of the parent. A good example is the different type of blood cells, red blood cells that determine ABO blood grouping into the human being. ABO blood group are controlled by gene I, isoagglutinogen. The plasma membrane of red blood cells has some sugar polymers that protrude from the surface, and the kind of sugar is controlled by the gene I. Over the plasma membrane of RBC, what kind of a sugars are protruding? It is decided by gene I. I gene has three alleles, IA, IB and I0. IA and IB are dominant, I0 is a recessive form. So that means when allele IA present and IB present, they produce a slightly different form of the sugars, while I0 will not produce any kind of sugar over the surface of RBC. So in this manner, because human beings are diploid organisms, each person will possess any of the two of the three alleles. IA, IB are completely dominant over the I. In other words, when IA and I0 were present, only IA will express. When IB and I0 are present, only IB will express. But when IA and IB are present together, both will express their own types of sugars. This is because of the codominance. Hence, red blood cells have both A and B type of sugars. Since there are three different type of allele, there are 6 different combinations of these three alleles are possible. You know, in the table it is shown. IA, IB, IA-I0, IA-IB, IB-IB, IB-I0 and I0-I0. These are the 6 different type of genotype that are possible. Homozygous A, AB, heterozygous A, homozygous B, heterozygous B and homozygous recessive. So when IA, IB are present, blood group would be A. When IA, I0 present, then also blood group would be A. This is homozygous A, this is heterozygous A. This is B, homozygous B, heterozygous B. This is the case of codominance. Do you realize that the example of ABA blood grouping also provide a good example of multiple alleles? Whenever any gene has more than 2 alternative forms of the gene, means having more than 2 allelic forms, this condition is known as multiple allelism. As in case of blood grouping ABO, there are 3 alleles for the same locus, IA, IB and I0, means more than 2, so this condition is a multiple allelism. Here you can see that there are more than 2, means 3 alleles governing to the same character. Since an individual can carry only 2, an individual can carry maximum 2 alleles, so multiple alleles can be found only when a population gets studied. Means in case of multiple allelism, single gamete or an individual are not enough to study all more than 2 alleles. To study more than 2 alleles, we need to study the population. So multiple alleles are always studied at the population level. Occasionally, a single gene may produce more than one product, means more than one effect. For example, starch synthesis in pea seeds is controlled by a gene, and we are denoting this gene as a B gene that has 2 allelic forms, capital B and small b. Starch is synthesized effectively by the capital B homozygous, means large size starch grain. Therefore, large size starch grains are formed. In contrast, if small b homozygous recessive is present, lesser efficiency of the starch synthesis we will notice. So, smaller starch grain will arise. Capital B is producing large sized starch grains, while small b is producing small sized starch grains. After maturation of these seeds, capital B seeds are round, means those plants which are producing large sized starch grain, they are producing round seed, and small b seeds are wrinkled. It means those which are producing small size starch grain, they are wrinkled. So heterozygote, now condition is that heterozygotes are producing round seed. It means from the shape of the seed point of view, capital B as well as capital B, small b, both are producing round seed, means dominance we are watching, and small b producing wrinkled seed. Yani in terms of the shape of seed, this character is showing dominance. But when we talk about size of the starch grain, so we must keep this thing in our mind, that the starch grain produced are of intermediate size in case of capital B, small b condition. It means if starch grain size is considered, as the phenotype, then from this angle, the allele showing incomplete dominance. Means same gene showing more than one effect. So this is condition called pleiotropic condition. In this pleiotropic condition, for one phenotype, this gene is showing dominance, and for the another phenotype, this gene is showing incomplete dominance. It means dominance is not an autonomous property of the gene. It depends on the gene, it depends on the product of the gene, as well as it depends on the selection of the phenotype. To which phenotype you are selecting? If suppose I am selecting to the starch grain size, I am watching incomplete dominance, but if I am selecting shape of the seed, I am watching a dominance. So therefore, dominance is not an autonomous property, autonomous feature of the gene or the product that it has information. Dominance depends on as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine in case of when more than one phenotype influenced by the same gene. It is a most important paragraph that you should keep in your mind. Dominance is not an autonomous property of the gene. It depends on the gene, it depends on the production of a phenotype from this gene, and it also depends on the selection of the phenotype that we choose to examine. Now, inheritance of the two genes. Mendel also worked with the with and crossed pea plant after considering two characters. So you know this is called dihybrid cross. So Mendel found that the seeds resulting from the crossing of a parent, he selected seeds with the yellow color and round, both are dominant, and the green color and wrinkled shape as the recessive character. Means the cross is going on, yellow seed with round shape crossed with green seed with wrinkled appearance. And when he did this cross, he found that the seeds resulting from the crossing of the parent, means F1, had a yellow color and they all are round. Means only dominant phenotype express. Here can you tell which of the character in the pair yellow green and the round wrinkle shape was dominant? Of course we can say yellow is dominant over the green, round is dominant over the wrinkled. Thus yellow color was dominant over the green color and round shape was dominant over the wrinkled. These results were identical to those that we found into the F1 into the monohybrid crosses. Yes. Let us use the genotypic symbols Y for the dominant yellow and small y for the green, R for the dominant character means round and small r for the wrinkled. It means parents are capital R, capital R, capital Y, small r, small y, small y. So F1 generation all members are heterozygous, capital R, capital Y, small y. When these are allowed to self-pollinate, when Mendel self-hybridized to the F1 plant, he found that the 3/4 of the F2 plant has the yellow seed and 1/4 has the green seed. Similarly, 3/4 have the round shape and 1/4 have the wrinkled seed. It means likewise. Capital R, capital Y, round yellow, wrinkled green. F1 generation, all plants are round yellow. Selfing, so it will produce now 4 types of gametes. Capital R, capital Y, capital R, small r, capital Y, small r, small y. Same female gametes also. When we see this checkerboard, what we found? That 9 members are round seed with yellow color, 3 members are round seed with green color, 3 members are wrinkled seed with yellow color, and 1 member is a wrinkled seed with green color. It means 9 plus 3, 12 out of the 16 are round, 3 plus 1, 4 are wrinkled. Similarly, 9 plus 3, 12 are yellow and 4 are green. It means round is to wrinkle, 12 is to 4, yellow is to green, 12 is to 4, means 3 is to 1. Means both of the character are following their monohybrid ratio. It means when we are considering 2 character together, each character is showing their monohybrid Mendelian inheritance. It means if 2 characters, inheritance of the 2 characters study simultaneously, one character, inheritance of the one character not influenced by the inheritance of another character. It means both of the character will exhibit in their typical monohybrid manner. This is called law of independent assortment. Means inheritance of one character is independent of inheritance of another character. The law of independent assortment is here shown here, that the yellow is to green, 3 is to 1, round is to wrinkled, 3 is to 1. So round 9 is to 3 is to 3 is to 1. It is indicating that the dihybrid cross or monohybrid cross are simply the number of character considering for the study of inheritance are increasing, but each character is showing their independent inheritance in their typical monohybrid manner. Like law of segregation, law of independent assortment also has its cytological basis, and that is also in anaphase 1. You can say anaphase 1 is the basis of law of segregation as well as law of independent assortment. Then chromosomal theory of inheritance. Mendel published his work on inheritance of character in 1865. From 1856 to 1863 he did work, but for several reasons it remained unrecognized till 1900. What are these reasons? Reason number 1, the communication in those days was not easy. Second reason, his concept of the gene as a stable and discrete unit that control to the expression of trait or allele which do not blend with each other was not accepted by his contemporaries. Yani his idea of the gene was not accepted. Third reason, Mendel's approach of using mathematics to explain the biological phenomena was totally new and unacceptable for the others. And finally, though the Mendel work suggested that factors were a discrete unit, but he couldn't provide any physical proof of the existence of factor that they are controlling. The 4 reasons are expressed here that are the reason why the Mendelian work remained hidden for approximately 35 years. So Mendelian genetics in this context is very important, but it also need to be explained on the basis of certain kind of a physical and chemical proof, and to explain that some research work further after 1900 started. One of the such attempt was the chromosomal theory of inheritance. In 1900, 3 scientists, Hugo de Vries, Carl Correns and Schermark, independently discovered Mendel's result on the inheritance of character. Also by the time, due to the advancement of the microscopy were taking place, scientists were able to carefully observe the cell divisions. This led to the discovery of the structure in the nucleus that appeared to be double and divide just before each cell division. These are known as chromosomes. By the 1902, the chromosome movement during the meiosis had been worked out. Walter Sultan and the Theodore Boveri noted that the behavior of the chromosome movement, noted that the behavior of the chromosome was parallel to the behavior of, very important line, that the behavior of chromosomes was parallel to the behavior of the genes, and used the chromosomal movement to explain the Mendel's law of inheritance. Means whatever the things said by Mendel for the factors, that the factors occur in pairs, that the factors segregate at the time of gamete formation. Similar behavior Sutton and Boveri observed about the chromosome, that the chromosome occur in pairs, that the chromosome segregate at the time of gamete formation, that the segregation of the chromosome is independent of the one another. Recall that you have studied the behavior of chromosome during the mitosis and during meiosis. We know pretty well. The two alleles of the gene pair are located on the homologous chromosomes on the particular site. So it is indicating that why factors are present in pair? Because chromosomes are present in pair. Why factors segregate? Because at the time of anaphase 1, homologous chromosome becomes separate from each other. Why segregation of the one factor is independent of the another factor? Because factors were located on the different chromosomes. It means who actually going from one generation to another generation. Chromosomes are going from the one generation to another generation. And because genes are present on chromosome, so whatever the chromosome will do, gene will also do. So genes occur in pair because chromosomes were in a pair. Genes segregate because chromosomes segregate. Segregation of the factors is independent because segregation of the chromosomes were independent. It means the thing that so far by Mendel explaining regarding to the factor seems to be true about the behavior of chromosome also. It means genes are located on chromosome. It was the finding come out after the efforts made by the Sutton and Boveri, and this is called chromosomal basis of inheritance. So this paragraph, this line is very important. You have to distinguish that which column is a gene and which column is a chromosome. Occur in pairs, both occur in pairs, means gene as well as chromosome, means from the point A, point 1, we cannot conclude. Segregate at the time of gamete formation, such a manner that each gamete receive only one pair. Segregate at the time of gamete formation and only one of the each pair is transmitted to a gamete. It is also equally applicable for the chromosome and gene. Independent pairs segregate independent of another. Yes, this is also true for both gene and chromosome. One pair segregate independent of another. This is always true for the chromosome, but this is not always true for the genes, because genes may be linked together. It means column B is always true for chromosome, but is not always true for the genes. But column A is always true for chromosome as well as always true for the gene. So it means we should consider column A as a gene and we should consider column B as a chromosome. So column A for the gene and column B for the chromosomes. Here it is shown in form of the independent assortment. Very important line that you have to keep in mind. During anaphase 1 of the meiosis, means anaphase, independent assortments basis is exhibiting. Sutton and Boveri argued that the pairing and separation of the pair of chromosome would lead to the segregation of the pair of factors that they carried. Very important line. Later on, Sutton united the knowledge of the chromosomal segregation with the Mendelian principle, and that is now it is known as chromosomal theory of inheritance. Following this synthesis of the ideas, experimental verification, very important line, experimental verification of the chromosomal theory of inheritance was given by Thomas Hunt Morgan. He did work on the Drosophila melanogaster, Why melanogaster means Drosophila? It seems to be very good material. Look, very suitable for the studies. Why Drosophila is very suitable? Because they could be grow on simple synthetic medium. Reason number 1. Reason number 2, they complete their life cycle. They complete their life cycle in about 2 weeks, and single mating can produce a large number of progeny of the flies. So because they can be grow easily on the synthetic medium, one of the reason, because they are producing large number of progeny after each mating, and because they have a life cycle short about of 2 weeks, that is also it is very important. Then linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila. The crosses were similar to the dihybrid crosses carried out by the Mendel. For example, Morgan hybridized yellow body white-eyed female with the brown body red-eyed males and intercrossed their progenies. He observed that the two genes do not segregate independently. Two genes do not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio. As the ratio disturbed from the 9:3:3:1, it means it is not independent assortment. Morgan and his group knew that the genes were located on the X chromosome. Which genes? Genes for the yellow body white eye and genes for the brown body red eyes were located on the X chromosome, and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combinations, was much more higher than the non-parental types. Means parental combinations were arising more than the recombinant combinations, than the non-parental combinations. Morgan attributed this due to the physical association or linkage of the two genes. Physical association of the two genes is the linkage, and he coined the term linkage. He coined the term linkage to describe the physical association of the gene on a chromosome, and the term recombination to describe the generation of the non-parental type. Means creation, maintaining of the more number of parental type, he is calling it linkage, and creation of the new forms, means the non-parental form, he called the recombinants. Morgan and his colleague also found that even when genes were grouped on the same chromosome, some of the genes were very tightly linked, means showing least recombination, while others were loosely linked, it means they are showing relatively more number of recombinations. For example, he found that the genes for the white and yellow body, white and yellow were very tightly linked and showed into the 1.3% of the recombinants. See the cross. In this cross, if you observe, what we are finding, that the yellow body white eye, that the yellow body white-eyed female when crossed with the red body, brown body red eyes male, in F1 generation, all the female Drosophila are brown body red eyed, means wild type, and all the males were yellow body white eyes, yani like mother. When they were intercrossed together, so wild means brown body red eye and yellow body white eyed obtain 98.7%, yani parental type, while brown body with white eye and yellow body with the red eyes, means recombinants, obtain just 1.3%. It means body color and eye color genes were very much linked together. In second cross, what you observe, that the white eyes with miniature wings, recessive character, crossed with red eyes and large sized wing, means wild type character. So all F1 females were red eyed with large wing, and all male progeny was white eye with miniature wings. When they are allowed to do interbreeding, what he found, that the red eye large wing and white eye miniature wing यानी parental combinations are obtaining 62.8%, while recombinant types means eye color red miniature wing and eye color white wing large, both of the recombinant that he obtained was just 37.2%. It means that the here parental combinations are arising 62% as compared to the earlier experiment where parental combinations were 98%. It indicates what? That here in the cross number A, cross A there is a more linkage, and in cross B there is a relatively less linkage. So you should keep this thing also in your mind, that this process of the crossing over is variable, or you can say this process of the linkage is variable. How far these genes are located from each other accordingly. Here these two genes were present very close, so they were showing more linkage. Here they were relatively distant, so they were showing relatively more number of recombinants. In the linkage and recombination, one more thing that we should keep in your mind, that Morgan's student Alfred Sturtevant used this frequency of the recombination between the gene pairs on the same chromosome as a measure of the distance between the genes, and on the basis of which they prepare gene map or the chromosomal map. Means more the distance between the gene means more the crossing over, means more the number of recombinant. So 1% recombination frequency they assumed to be the 1 unit distance between the gene. So this idea of the Sturtevant to use the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance between the gene and mapped their position on the chromosome, that is the basis of the genetic map. Today genetic maps are extensively used as a starting point into sequencing of the whole genome, as was done into the case of human genome sequencing. Polygenic inheritance. So far what we observe, that the one character controlled by one gene, but whenever one character controlled by more than one gene, means more than two allelic forms, so then this kind of inheritance called polygenic inheritance. Mendel studied mainly described that those traits that have distinct alternative forms, such as the flower color, whether it is a purple or white. But if you look around, you will find there are many such kind of traits which are not so distinct, means which are not just in the two extremities. For example, in case of human being, we do not just say that the tall and short people as two distinct alternatives, but the whole range of the possible heights are possible. Such traits are generally controlled by 3 or more genes and thus are known as polygenic trait. Yani human height is a polygenic character because it is controlled by more than 2 allelic forms, means 3 or more allelic forms. Not just only human height, human skin color is also not confined in between only black and white. There are also many shades. Whenever any character is not just confined only up to the two extremities, instead it has a variety of range of the trait between the two extremities, then such type of a trait are definitely going to be controlled by more than 2 allelic forms, means 3 or more allelic forms, and that is called polygenic inheritance. In human being, skin color is the another classical example of this. In polygenic trait, phenotype reflect to the contribution of the each allele. The effect of the each allele is additive, means suppose I have capital A, capital A, capital B, capital B, capital C, capital C. So these 6 dominant alleles showing their effect in an additive manner. But if I suppose have capital A, capital A, capital B, but capital C and small c, means one C is missing, capital C is missing. So my phenotypic expression would be relatively less than the earlier condition. So the effect of the each dominant allele is additive. To understand this better, let us example of the 3 genes ABC controlling to the human skin color. When all 3 dominant alleles are responsible for the darkest skin color and the recessive forms had yani small a, small b, small c have the light skin color. The genotype with the all dominant allele, means capital A, capital A, capital B, capital B, capital C will have the darkest skin color, means negro, and the small a, small a, small b, small c, small c will have the lightest skin color, means Caucasoid. As expected, the genotype with the 3 dominant allele and the 3 recessive allele will have an intermediate skin color, means capital A, capital B, small b, capital C, small c. And then the character will fluctuate on either of the side. In this manner, the number of each type of allele in the genotype would be determine the darkness or the lightness of the skin of an individual. Later on, there is one more aspect, that is pleiotropy. We have so far studied just a reverse of multiple allelism. There multiple allelism, what was going on? One character, more than 2 gene or the allelic forms. Here there what we are watching, one gene pair regulating to the more than one phenotype. I can say just reverse of polygenic inheritance. Character 1, more than one gene. Here gene 1, more than one character. So far we have affected that the effect of a gene on a single phenotype. There are, however, some instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called pleiotropic gene. The underlying mechanism for the pleiotropy in most cases is the effect of a gene on the metabolic pathway which contribute towards the different diverse phenotypes. One of the well-known example is a phenylketonuria, which occurs into the human being. The disease is caused by a mutation into the gene that code for the enzyme phenylalanine hydroxylase. Phenylketonuria is a disease caused due to the deficiency of enzyme phenylalanine hydroxylase. This manifestation, what effect comes? Expression of the character, this manifests itself through the phenotypic expressions characterized by the mental retardation will appear, one of the phenotype. Reduction in the hair and skin pigmentation is the another effect. Means the same gene is controlling to the mental retardation as well as hair color as well as skin color. So multiple phenotypes controlling by one gene, that means the pleiotropism. We have already discussed, if you remember, gene B that was controlling to the shape of the seed in pea plant, round and wrinkled, as well as that was controlling to the size of the starch grain, large, intermediate and small. Means in pea plant, that gene was also pleiotropic gene. So this is all about the genetics, principle of inheritance portion. You complete it from the NCERT thoroughly. It is a very important and significant chapter. Do practice of the numericals also, which are given to your exercises. Thank you very much. Wish you all the very best.